Welcome to Biotech Stage. It's a stage where we meet with biotech professionals from the globe to discuss something very important, something very hot, and something very interesting in the biotech field. Today's topic is also very interesting and very important. But before I reveal that topic, let me give you one scenario. A person goes to pharmacy or a drugstore to buy a particular medicine for his, let's say, diabetes or blood pressure or simple pain medication. He buys a particular drug from that drugstore from a company that he is a little bit familiar with. When he buys, he does not think twice whether the product is pure, whether the drug is stable. If it says it is good for next 2 years, he doesn't question at all. He assumes that the drug is going to be good for next 2 years or 6 months or whatever time period it says on the label. And most importantly, he doesn't think at all whether a drug or that particular medicine is going to work as it says on the label. Why? Because he knows that the company that he is buying it from has done enough work through its researchers, through its technicians in the lab to make sure that the drug is not only pure, it is stable and it is going to work according to what it says on the label. And same thing is true for the biologics. And actually, for biologics, it is even more complicated because biologics are large molecule versus a smaller molecule, and that's a different discussion. Maybe we can discuss that on some other point of time. But today we are going to discuss when somebody buys a biologic medicine from the store, from the medicine shop, what are the different things that are done to give surety that the product is stable, product is pure, and product is going to work as per specifications, as per the label. And to discuss all that, we have in our studio or on our stage Dr. Saurav Majumdar, who has been working at Agile Biosciences. So welcome, Dr. Saurav Majumdar, on this show.
Thank you very much, Dr. Arun. I'm really grateful that you invite me and provide me a platform to talk on this topic. Really, it will be a very helpful information if I provide it, and this is very encouraging.
Thank you, Dr. Saurabh. So, first thing which I want to know is when it comes to biologics, I understand there are different types of testing that could be done, whether it is a product development or whether it is a analysis of the final product to make sure that drug is safe, drug is the structure and function relationship is done, stability is done, and for biologics, the immunogenicity part that is also important. And India is big on biosimilars, and if the biosimilars are being made the comparability studies to make sure that it is similar to the original molecule, all those things, and most importantly the regulatory expectations, that is also done. So if you could please tell us something about different types of biological characterizations that are being done on proteins and the vaccines.
It's a very good questions. That really it's a very unknown topic for everyone. When a drug is developing, so from the clone to market authorizations, there is a life cycle. And in the life cycle, if you go, so in the drug development way, characterizations is playing a very important role. And characterization is not only the superficial part of the characterizations, the molecule, which is a very small molecular weight like insulin or parathyroid hormone, to a molecule like MAP. And these series of molecule from the low molecular weight compound to high molecular weight compound, these all molecule will be characterized thoroughly in order to check the safety, efficacy, and purity of the drug product. So this kind of things will be done in a details investigations or the study that is called the bioequivalence study as compared to the reference product. If the reference product is available in the market, for I am talking more focused on the biosimilar. So in that case, that primary level characterization will be done based on the amino acid sequencing, amino acid composition analysis of that particular protein, disulfide bridges analysis, peptide mass fingerprinting, aggregate analysis, protein concentrations, and also it will show a variety of experiment between the receptor and the drug binding study by using the SPR or BLI platform. So SPR and BLI platform is nothing but your surface plasma resonance. So Biacore T2, which is currently mostly the going to obsolete, but the Biacore has a system where we can do this analysis between the receptor versus drug in a fluid mechanics mechanism. And BLI is called the biolayer informatory. So this biolayer informatory is the principle for the octet. So this octet is doing the same thing like SPR, and they are going to find it out the on rate, off rate, and KD value for this molecule when we compare it against the receptor versus particular drugs interactions. Every time animal study is not feasible because it is quite expensive, but the same thing you can mimic in vitro system, and the way drug is working on in vivo, the same thing you can mimic in vitro system. Before that, as I initiated with the primary level characterizations, so in primary level characterization, if you see the protein amino acid sequence is very important, because this protein amino acid sequence, intact mass analysis, subunit mass analysis, it is very, very important. Why? Because if the molecule is a two different type of molecules are there. First one is the glycosylated molecule, another one is the non-glycosylated protein. If the protein is generated in the microbial sources, for example, insulin, the glycosylation part is not there, as if since the post-translational modification will not happen there in bacterial cells. But when you are talking about something called the MAB is a very critical molecule where this glycan is playing a very, very important role. And this glycan is also give a serious input between the receptor and particular drug interactions. If the molecule is not properly glycosylated, that it will not show the receptor and drug interactions the way it will do for the reference product. So in parallel, reference product versus your product will be analyzed under identical conditions to provide an information that the binding affinity between the receptor and your drug is a very similar and very close to the reference product. But before that, the glycan profiling is also very important, as I said, because we are doing the glycan profiling because when we have started the batch from the small scale to higher scale, like 250 ml taking flux to a fermenter, like 10,000 liter fermenter, in that case, when the molecule, the batch size is increasing, possibly the glycan profiling of that particular molecule will change.
Okay.
At the same time, it will change its acidic and basic variants. And because there is a chance of oxidations, there is a chance of deamidations in according to the charge variant profile, and glycosylated profile is a very important part for the molecule characterization, because you have to show the way you are doing for the 10-liter batches or 250 ml, the smaller one versus the molecule with the 10,000 liter when it is the big scale. So your molecular entity will remain same.
Okay.
So in this case, actually we are deglycosylated the protein, because we are using the N terminal in a linked glycan. It's not N terminal, it's an N linked glycan. So we are digesting with the PNGs, and then we isolate the glycan and protein. We isolate completely a helichromatography part, that is the hydrophobic interaction liquid chromatography, where the protein is isolated and glycan part will isolated. Then we dissolve the glycan and tag a fluorescence tag, and then we analyzed in the UPLC or HPLC by using the effluent detector. So these are the part when you will see a glycosylation profile. So there is a multiple glycosylation moieties are there. If you see the background of that particular, if you just think about the processing of the post-translational modifications, actually the molecule, when it is processed in a CHO cell or when it is going through a host cell from here to Golgi, and Golgi, trans Golgi to cis Golgi. So like this way it will travel. At the end of the release of that particular molecule from the Golgi, that time it will the glycosylation moiety will be properly formed.
Mhm.
And in the glycosylation part, the background or the back, there is a two different thing in the glycosylation. One is the core area, another is the terminal area of glycosylation.
Okay.
Usually in core area of glycosylation, mannose are there. But when it is called something called when the molecule is processing inside the cell from here to Golgi, from cis Golgi to trans Golgi, and then it will come out, that time the terminal glycosylation part will be there. In addition to the glycan moiety, there is also another sugar molecule called sialic acid will be attached along with the glycan moiety. Why this is very important? Mannosylation, effucosylation, and galactosylations, these are the three different parameters usually we measure. If the mannosylation is very high, there is a chance of early clearance from the body of that molecule. If you inject it, it will clear from your the bloodstream because the shelf life of that particular molecule will be very less.
Mhm.
And at the same time, it will not show similar kind of biological activity compared to your reference product. Similarly, in case of galactosylation and effucosylation, it will give a strong impact on ADCC and CDC activity. So these are the part which is controlling the quality control parameter of that particular molecule. So that's why this parameter is known as a critical quality attributes.
Okay.
That is known as a critical quality attributes and CQA of that particular molecule as a features for the characterization purpose. So
Okay.
these are in similar way. When the molecule is oxidation, when the molecule is deamidated. If a molecule is oxidation, then the methionine residues or the tyrosine residues, these molecule is going to affect. Because in the sequence, if the methionine is there, this methionine will convert in the form of methionine sulfone and sulfoxide. And that will change the orientation of the molecule. This orientation of the molecule will be differ from its original native conformation, and it is going to show impact with the receptor binding study when you will do the receptor binding study by using the SPR and BLI. So a result which you are getting the more acidic variant, so that is going to impact the physiological activity of the molecule. So
Okay.
that's why this reference product, it will be compared along with your in-house product in order to attest that the molecule is biosimilar. Now the
Mhm.
same thing, if it is I'm talking about something called the MAP. If you think about the PTH hormone, that is the parathyroid hormone, is also one of the examples. If you
Mhm.
see, this molecule is 8 kD molecule. Around 32 amino acids are there. So in that case, 8 and 18 position of the methionine residues are sequentially oxidized. And these oxidation, when you do, if you do the receptor binding study against the control sample, you will see the activity of the molecule was diminished.
Mhm.
And that is the reason the oxidation, in similar way the deamidations itself is telling asparagine to aspartic acid, glutamine to glutamic acid. So removal of NH2 group and converting in the form of aspartic acid or glutamic acid, it will similar way like oxidation, it will change the native structure of the particular protein. And this protein molecule is going to impact on the receptor binding study a very strong impact, and as a result the activity of the molecule will be progressively down. Even though you have increased the batch size, you have produced the molecule, but when you will check the activity of that particular molecule, this activity of the molecule may go down if you don't provide the exact characterization. And through a comparability study between the 10 liter to 2000 liter or 10,000 liter batch under identical platform in respect to your reference product, so that time you will come to know that molecule is not active. So that
Mhm.
is the scientific background. At
Mhm.
the same time, we are also, this is the primary level characterization I was talking. Now I will switch over, because you know the protein is just like a backbone of these amino acids. Now it will fold it. Now in the folding structure, when it form a secondary structure, so that secondary structure will be analyzed by the CD. In CD analysis, we are scanning the molecule at far UV wavelength at 190 nanometer to 260, where alpha helix, beta turn, percentage of turn, and random coiling, these four parameters will be recorded. The alpha helix and beta sheet or turn, if it is changing compared to your reference product, that's mean the folding process will be changed. And amino acid interaction between a particular domain with your receptor, if it is changed, then it will subsequently change the biological activity of that particular molecule. So that is the reason we are doing the secondary structure analysis by CD, and also at the same time we are doing also FTIR. So CD is there, FTIR is also there. FTIR is denoting the amide 1 and amide 2 peak. So amide 1 is nothing but your alpha helix, beta helix turn in all together, it will give you a picture. So that is also encouraging the structure of that particular molecule. In near UV, the tertiary structure will be evaluated by using the CD analysis, where the aromatic group of amino acid like phenylalanine, tryptophan, and tyrosine, these amino acids are playing a very important role in the protein native structure conformation. If it is on the surface or if it is in the buried conditions inside the molecule, that is also need to address when you are going to confirm the structure of that particular molecule is similar to your reference product.
Okay. Okay.
And later on, we are also doing the tertiary structure analysis. In tertiary structure analysis, we are exciting the molecule at 280 and 295 in order to observe that the extrinsic fluorescence or intrinsic fluorescence study, these tryptophan and tyrosine, both the molecules are on the surface or in the buried side, it will show a lambda max absorbance, and that is indicating the reference product versus your molecule is exactly the same orientation, because you have excited at 280 and 295 and take a emission spectra from 320 to 400. So this will tell you the molecule is similar to that. Apart
Okay.
from that, also in Agile Bioscience, we are doing a very important study that is called the AOC. So AOC will be tell
And
you,
AOC stands for AOC.
AOC stands for analytical ultracentrifuge.
Okay.
This analytical ultracentrifuge, without disturbing the molecule, we are analyzing the thing to understand the quaternary structure of the protein, and in the solution it is contain any high molecular weight or low molecular weight component along with the native conformation is present or not. So sec mols, sec HPLC, AOC, these will tell you the overall picture of the aggregations,
Mm-hmm.
because aggregation is also a part of protein chemistry, which denote as a very important factor for the deterioration of the protein or the stability of the protein.
Mm-hmm.
So in this case, a protein, if it is misfolded or that protein molecule is open is hydrophobic patches, so it will interact with the other molecule and it will form a aggregate, and that aggregate is maybe stable or it will be dissociated or reversible aggregate or irreversible aggregate. If
Okay.
this kind of molecule binding in between the two structural entity is there, then through the disulfide linkage, then it is denoted as a covalent dimer.
Mm-hmm.
If it is not linked by the disulfide linkage by means of hydrogen bond or a bond which is very loosely associated with these two particular protein entity, that is considering as a reversible, because it is formed in the aggregate but later on it will be dissociated anyway in the form of monomer conditions. It is
Okay.
not a stable aggregates. So these kind of parameter will be evaluated by using this kind of system, and it will show you the aggregation study. Apart from that also, we are also doing one analysis called the nano-DSC. So nano-DSC will tell you the TM value of that particular molecule. If the protein is deteriorated in case of oxidation, deamidation, or aggregations, the TM value, or the temperature of melting point of that particular protein will be changed. Now,
Mhm.
most of the thing is that if you really understood the bioenergetics part of that particular molecule in a folded form, if you remember the Gibbs free energy, that is your delta G equal to delta H minus T delta S. The folding of particular protein, if it is there, so that folding is very compactness of that particular protein, where the entropy is very less. But when the protein is from the unfolding, so that randomness will increase, and that randomness will increase the your entropy structure, and that particular changes will be denoted from the DSC point of view, with the TM value will be changed from your control sample, and it will denote that your sample is start deteriorating. So
Mhm.
these will denote the stability of that particular protein under identical condition whenever you are compared with the reference product or reference standard. These are the overall things.
Okay. So thank you so much. This is a lot of technical details, but obviously this is very important. I think you mentioned about 12 or 13 or 14 different types of tests for
Yes,
primary
yes.
structure, secondary and tertiary and quaternary structure to make sure that the protein or the particular drug molecule that is going to the market is stable, is safe, and it is going to work as the instructions, as per the label or as it is supposed to work. And just for our audience, it was a very highly technical talk, but I just wanted to mention that Dr. Majumdar mentioned bacterial cells and mammalian cells. So just for our audience that is not that much of technical, I want to let them know that the proteins can be made in bacterial cells and as well as the same time in the mammalian cells. And the most of the mammalian cells at this times are Chinese hamster ovary, CHO cells, which is an acronym basically. And he also mentioned that CD, you mentioned CD, and that is circular dichromism, right?
Yes, yes, that is circular dichroism. Correct.
Okay. So, Dr. Majumdar, the thing which I wanted to mention is all these tests, if you could just tell us how long does it take I mean, once the protein is made, how long does it take to do all those 14, 15 different types of tests before it passes the QC?
See, this characterization actually include the stability indicating test, release test, and all together. Because
Hmm.
characterization, if you see, it's a big river, and
Hmm.
so these are the different ponds which is there in the river or different entity. Because in release test, all the test of characterization is not in.
But typically, the reason I'm sorry, the reason I'm asking this is when the protein is made, typically it takes maybe 2 weeks, 3 weeks to make. The culture is done for 2, 3 weeks
Yes,
typically.
yes. Around about 2 months it is required to do the
Okay.
full characterization study.
So it takes even longer to characterize, to make, maybe take 1 month or one and a half
Yes,
months and then an additional two
2
months.
months, around
Oh,
2 months period of time with some buffer line and failure and repeat analysis,
okay.
because we need
Okay.
to confirm the structure. So it is required when you are comparing.
And I guess that's the reason the cost of biologics is so high, because not only the
Yes.
manufacturing part is expensive, but also the purification and the characterization part. So one thing that I also wanted to ask on this is, I mean, obviously it requires a lot of, I mean, instruments and tools. Are those tools, are those reagents, are those instruments, are those machines available all in India, or some of the work has to be outsourced?
See, most of the company recently they have procured,
Okay.
but most of the companies procured. But a CRO like Agile Biosciences, there is many CROs are there. But so far as my understanding and that my previous experience, most of the CROs are not set up these entire biophysical characterization under the same roof,
Mm-hmm.
which Agile did.
Okay.
Because we have focused the characterization from primary to quaternary structure of the particular
Mm-hmm.
protein by using the multiple analytical method or the
Mm-hmm.
various analytical method. So that thing is very important, because it is not possible for any company to to match the timeline and to regulatory submissions by doing all the test. So that's why they are doing the test from the outsource the test from the CROs.
Okay.
So CRO is playing very important role, and they are taking the molecule, they are doing these exercises, and they are producing the record. They are recorded the results in terms of method development, qualifications, and the report preparations,
Okay.
which is required from for the submission purpose. So the way we follow, actually, because in CRO we are not a product oriented. Please
Mm-hmm.
understand this part. It's not a specific product we are manufacturing here. We are
Mm-hmm.
receiving the product from the multiple companies.
Mm-hmm.
So the nature of the molecule will be different.
Mm-hmm.
Although the methodology we are using, possibly the 90% similarities are there from one molecule to another molecule. When you do
Mm-hmm.
one map, you can do another map. But
Mm-hmm.
still, the method development part is a very by default it is included, and the qualification of that particular method or the partial qualification, it will attest that the methodology we have established for particular dedicated product, that methodology is consistent,
Okay.
the
Okay.
result is consistent. So that
Okay.
methodology we are using for that. So like
Okay.
this way we are doing the work in Agile Biosciences, and this is divided into three different segments. First
Mm-hmm.
of all, the method development or the feasibility testing or the proof of concept study. It
Mm-hmm.
depends. The clients is required, because the client is, it is the as per understanding with the client that what the requirement of the client. If they
Mm-hmm.
want it, we are doing all this activity, but we have to do it at initial stage. Then we have started the characterization part of the work, and in characterization we are working this method by using the method to characterize this molecule in full dimension.
Okay. Okay. It is great to know that your company can do basically all four types of tests of primary, secondary, tertiary, and quaternary structure for the proteins. So basically, that's all the things under one roof. So that's great to know. One other thing which I wanted to find out from you is I understand biologics are expensive because of such tests. So when it
Yeah.
comes to, I mean, what percent of the cost is towards that these types of tests?
Yeah, 10% of means around minimum 5% of the cost,
Okay.
because it depends on the CRO also. So
Okay.
that how they are defining their cost, it vary from one CRO to another CRO.
Mm-hmm.
But since it's a one-time activity for the before toxicological data submissions and at the same time in the product life cycle,
Mm-hmm.
and later on it will come again to the CRO when the phase one, phase two, phase three will come in the picture. For
Mm-hmm.
biosimilar, it is phase one and phase two. That comparability study will be carried out again through the characterizations method.
Mm-hmm. Mm-hmm. Okay.
So these things is matters, and that will evaluate the cost.
Yeah. Okay. But again, it is great to know that Agile Biosciences can do all the tests because India is focusing a lot on Make in India efforts. And companies, if they can do all the work in India, I mean, within Agile Biosciences in Ahmedabad, so if they can, if all the work can be done within India, obviously that highlights and supports the efforts for Make in India objective. So that's great to know.
And
So.
some of the client is required, because we are doing this because most of the characterizations part is not required the qualifications, and
Mhm.
also a few people are doing the exercises so without any supervision of QA. It is not the
Mhm.
system like Agile Biosciences. So with Agile Biosciences, we have a system called the quality control system, and under purview of QA, the data
Mhm.
review will be done, check,
Mhm.
and then it
Mhm.
will be forwarded to the client. And under
Mhm.
GMP environment or the GLP practices, the recorded of result in the DRS, in the
Mhm.
method development, there is a particular format is there. where we are recording the result, and it always done under the purview of QA. So we have around 70 SOP to follow this. So if a person is required to do the analysis in the GLP norms, so as per the GLP norms, so we have that facility to address this molecule, and also we are doing this practice and recorded the result accordingly.
Okay, okay, okay.
And also we have a soft copy of the data, because maybe you are doing the characterizations and you have submitted 3 months, 4 months, 5 months later, you are asking me the data, because reviewer may have asked this kind of questions that they want to verify, because it is most of the thing is very suspicious. If it is in their observation, they can ask the raw data. So we have a system of backup,
Mm-hmm.
and we have also the audit with the various companies, and these are the well-known companies. And also we have audited the number of companies located in Hyderabad. Those are submitting the documents from Brazil and Canada. So these are the international market and US. So as per their norms also, we are submitting the documents. See,
Okay.
analyzing the part is okay, but when you are terms of the regulatory report submissions, so it's very, very crucial. See, this is not, I am saying this is not a academic part. When a person is doing the research and writing a paper, the matter will be over. For
Mm-hmm.
that, the core expertise is there. I always appreciate the researcher. Those are involved, because of them only we are getting so many information from the research article. But that focus will be more for the regulatory submissions, what their
Mm-hmm.
requirement. So according
Mm-hmm.
to the US FDA, EMA, TGA, these are the various agencies. There's places when you are going to send, that company was decided to send the molecules in Australia. So the TGA will ask the document. So you need to prepare the document as per the requirement of TGA, and we have to
Mm-hmm.
submit it with the zero observation till date. We have not got any kind of queries over there. So documentation part is very strong. We follow the QMS properly to obtain the document in a specialized way.
Thank you very much, Dr. Majumdar. This is a very important and talk, and thank you very much for giving such great technical details. Your talk, I mean, your information, I'm sure will be very valuable for the people who are very much interested in biophysical characterization of proteins, or they are working in QA, quality control, quality assurance, and also it will be very useful for the young scientists who are trying to enter into this field, and they want to understand how the proteins are characterized. I mean, you have given the complete overview and with all the details, so different techniques that are being used. So this will be very important for all the young scientists also. And also, I think it is going to be very useful for the people who are trying to apply for different jobs. Because I think a lot of people don't realize that it is one thing to say that I have done protein characterization, and it's a totally different effect when somebody says I have done the protein characterization that can be used for the secondary structure determination of that particular protein. So I think it will be very useful for different types of audience. So thank you very much again for your valuable time.
Thank you.
Thank you.